Okay, maybe I can slowly start with the introduction of this webinar. So welcome to this 35th webinar organized by Instruct in the series Structure Meets Function. So just a bit of a reminder or introduction for those that do not know about what Instruct-ERIC is. So we are the research infrastructure for structural biology in Europe. So we offer access to facilities all across Europe, offering services for integrated structural biologies, but we also offer other type of services to our scientific communities, such as training activities, internships for early career researcher, and R&D funding for small research projects to support our research community. So as I mentioned before, we have centers all across Europe. As you can see, we have 11 centers in Instruct that offer different services that are important for structural biology. So going from sample preparation with services such as crystallization, nanobody discovery, and protein production, to molecular biophysics analysis of samples with imaging services, molecular biophysics and mass spec services. And we also offer high-end 3D structure analysis services such as X-ray techniques, NMR, and EM services. So today, my pleasure is to introduce to you two speakers that have actually used the Instruct facilities via the ISIDORe project. So Instruct is part of this ISIDORe project that is supporting researchers working on infectious disease with a high risk of pandemic. So it was, of course, launched just after the COVID-19 pandemics, when we realized that we had to make more efforts in getting ready for this type of pandemic. So we had two emergency calls supporting the SARS-CoV-2 COVID-19 pandemic, and then the monkeypox epidemic that started soon after. And we had some calls that were more to support research on other pathogens that have a high risk of pandemic and that we need to study more in depth. So today we will present markers on long COVID that will be presented by George Vavougios, that is currently based in Cyprus.
Hi, Dia. Thank you very much. And thank you for having me. We're going to discuss about the serum proteomic and exosome tau overlap between Alzheimer's disease and the post-acute sequelae of long COVID, specifically the cognitive correlates between the two, which was an ISIDORe supported project and something that was only possible through Instruct and ISIDORe itself. Without further ado, there is, let's say, a minor conflict of interest, meaning that we're involved in further grants and development of this project, but nothing too nefarious. And the overall idea began from our clinical practice, where we still follow up on patients that had COVID-19 during the pandemic and subsequently, and blindly after 2 months. So even if they didn't report any symptoms, we followed them up with a comprehensive review of all the systems and with a specific focus on cognition. So, and of course, everyone that would be involved in that would have to provide informed consent for subsequent usage of their biological and other data. The comprehensive evaluation involved all aspects of the health, and since it was primarily let's say a respiratory medicine department, it was respiratory medicine and respirometry, but also several aspects of general health and cognition. And out of that data, prior to adding on proteomics, we had a pretty good idea early in the pandemic that most of them had some kind of memory impairment, and for some people it was, let's say, implicated with the overall consequences of COVID-19. So we had a general idea that COVID-19 had a unique, let's say, behavior as a generator of a cognitive phenotype. It really resembled other respiratory disease, but there were some people that resembled Alzheimer's disease, meaning mainly amnestic deficit, and we proved that as much as it can be proven via an analysis of eigenspace decomposition. It really just identifies patterns based on the neuropsychological data we provide. And we also identified some patterns with a respiration, meaning that there were some lasting deficits that didn't have to do with inflammation or active infection, but were a remodeling, if you will, of the respiratory system, at least on the executive part. And so we kickstarted some studies that aim to evaluate that. Covalent is the, let's say, the principal study that Instruct made possible, and the Cognate study, which is an affiliate, a spin-off, if you will, focused on exosomes, trying to figure out a brain-to-lung axis, or let's say an axis that focused on the brain. We always had a theory about type 1 interferon and how it can affect its tonicity, how its tonicity specifically can affect health and specifically cognition. And let's say, as a neurologist, I am a proponent of the Moore school, if you can call it that, in that we consider that neurodegeneration, viral, specifically infections, and trained immunity are intertwined, and mechanisms that are perturbed in all three possibly are so because they are close to their pathogenesis, to the pathogenesis of neurodegeneration. And we've been on record on the theory of the matter. So all that really is left is how to prove, if we can, via proteomics, that several months after an acute infection, you have lasting perturbators in your proteome, meaning that some proteins and some networks of proteins are dysfunctional, and that possibly points to, let's say, an overactive or aberrantly active innate immunity. And this hypothesis relies on the brain being informed of peripheral infection without an actual infectious agent being present in the brain. This is a model that when we began this study was probably on the fence, but now there are a lot of studies that I could quote, neurine or otherwise, that suggest that especially the hippocampy and other areas of the brain critical to cognition are informed of the, let's say, the tonicity of the peripheral interferon-1 tonicity, and potentially are disturbed by abrupt changes in it. And the contributors are the microbiome, as well as pathogens from every possible system. So what did we, let's say, specifically try to find? We tried to find whether the exosomal cargo and was deleterious. So for the first part, we tried to find out whether exosomes from the brain carried proteopathic seeds and had any differences between people who had cognitive deficits and those that didn't. And let's say the easy answer is that yes, they did. We found tau for the first time coming out from the brain and detected in the periphery. And that, let's say, tau, we're trying to find out whether it can kill or at least stress neurons, and therefore have a model to figure out who will suffer long-term consequences following COVID-19 and who will not. And so that this is where the exosome project is. So we had a marker, a pretty specific, let's say, marker of neuronal damage and a way for that damage to occur. But the real question is, and the question by which really proteomics can answer, is what's happening in the periphery. In 2024, we know that the periphery, especially in Alzheimer's, or at least the premonitory steps towards it, is as important, potentially as important, as the brain itself. A lot of peripheral processes that degrade amyloids occur in the periphery, and a lot of the genetic, let's say, burden of Alzheimer's disease come from immune genes and genes that have to do with infection, and we really don't know why. But what we did find is 42 proteins that are related to peripheral catabolism of macromolecules, and some of these molecules, 41 out of 42 of the proteins discovered by the proteomics experiment we performed thanks to ISIDORe, were related to serum proteomes and were pretty identical to some serum proteomes reported in Alzheimer's disease. Now, we analyzed that through our NIME workflow that essentially is designed to absorb differences in, well, pretty much anything we desire. But right now we wanted to compare these groups of people and these proteomic changes to control for the influence of multimorbidity, age, gender, and, well, and essentially compare infection status. And the proteins we arrived at, if you can, yeah, they appear. These proteins are not, let's say, random, and they're not newcomers or, let's say, unknown suspects. PZP and its partner A2 macroglobulin are a principal component of macromolecule catabolism in the periphery, and they're implicated in some of the mild cognitive impairment studies coming from the ADNI dataset, biggest dataset right now on dementia and cognitive impairment. So we have partners in crime. These two work together, and they are somewhat consistently found in people that are at the MCI stage or prodromal AD. And so as you can see with, and although we didn't do proteomics in extracellular vesicles, because they were separate projects, we did try to find retrospectively whether these proteins would be detectable in another study that used COVID-19 EVs. And we did find some known culprits, the 67 proteins that are even more, let's say, suspect on that. And if we take an unbiased look at the data, we'll see that the proteomic landscape of PASP and probably not only from our own study, but from other scientists that we, let's say, communicated our results with, is a continuum. So we find that innate immunity cascades and even humoral lipid metabolism and coagulation are perturbed for months, if we consider the continuum, for months after the infection. This can account for phenotypic variability that leads to reports of increased cases of stroke or rheumatic disease or cognitive impairment that we have. But specifically for Alzheimer's, so on its own is a pretty big finding. It shows that from a very humane side, we see that we are not well, may seem well, but definitely our proteomes aren't well, and the proteins going on circulating in our blood aren't, let's say, benign. And the other thing is that this looks very much like what we find in MCI and prodromal Alzheimer's disease proteomes. So this is, let's say, a very disconcerting fact. It doesn't have the ability to function as a mechanistic proof of one causing the other, but it overlaps with mechanisms that will make matters worse for all these things. So pretty much what we predicted, unfortunately, and for specific proteins, for interferon, the interferon axis, if you can say, if you can specifically focus on that, unfortunately, it's validated. And I will say not only from our own study, but we're certainly part of that. Okay, so studies that are already published, we're in the process of publishing our own, find the same molecules that we found in these people that had cognitive impairment following COVID-19. And as you can see in studies powered and specifically designed to study Alzheimer's, they also correlated these specific molecules, these specific proteins with neuropathology, or at least indirect evidence of neuropathology. And although we didn't go into extreme depth, mainly due to what we could do, we did detect and we did find overlap between this proteomic landscape and tau content. And another worrying, perhaps, prospect is that many of the mechanisms by which COVID-19 affects our proteomes might not be unique to COVID-19. So, and that might account why we find these things in COVID-19. I'd like to thank all the people who made it possible. So you may find me speaking, but it's probably all these people that made it happen. And last but not least, Ranjani that did all the mass spec experiments, and all the people at Leeds at ISIDORE brought me in contact with. Yeah, without further ado, thank you very much. I was very happy to be here.
Thank you so much, George, for a very interesting talk. If there are any questions, so maybe I will start to give time to ask other questions. So you mentioned at the end that it might be something found with other infectious diseases and COVID-19. So what are your proof? Are there some correlation that some people that have had specific type of infectious disease are more prone to neurodegenerative disease, and what type of symptoms or infectious disease would that relate to?
That's actually a very good question, Pauline, and thank you for that. The overall idea of type 1 interferon tonicity is that some of these pathogens, and COVID-19, SARS-CoV-2, are very capable and have molecular tools that perturb specific type 1 interferon interactors. So it's not really a matter of which pathogens do what, but probably how long do the effects on our own type 1 interferon tonicity last, and how long these effects are communicated to the brain. A very quick example that I can bring is that SARS-CoV-2 has, let's say, a dedicated relationship with alpha-synuclein, and it can cause aggregates. If you look at some ACS publications that are pretty distressing, but on its own, this, for instance, can't create disease. It can stress a system which, if it has, let's say, a genetic propensity, for instance, SNPs that are very common and very unlikely to cause on their own the disease, but likely to stress the system. So it's a multi-hit hypothesis. But our own, let's say, idea is that complement specifically, and probably let's say the duration of type 1 interferon dysregulation, along with a couple of tissue-specific events, governs the, I wouldn't call it penetrance, but let's say the manifestation of a phenotype or a disease.
Thanks. And did you see anything that would be variant-specific in how SARS-CoV affect the pathway, or it would be any SARS-CoV variant having?
Yeah, the worrying thing, if you will, is that, and a very simple, really, concept, if you think about it, is that for Alzheimer's disease, you need monocytes, and you need, let's say, free type 1 complement receptors, specifically C1R, which is the premier, let's say, mechanism by which peripheral amyloidosis is averted. So if you have a lasting dysregulation in complement alone, which COVID-19 does, to be fair, you sabotage the main mechanism by which our brain detoxifies itself peripherally from amyloids. So you don't really, you can say that you have Alzheimer's, okay, but you actually have a direct attack on the principle, at least as we know, peripheral mechanism of amyloid clearance. So this is something that COVID does very well. And based on the data we have, it does that, it continues to do that 6 months after the primary infection. It might be, though, that we don't have data for other pathogens for that long.
When you say that it does that even after 6 months post-infection, are you checking? Are you talking about patients without selecting for people that keep having symptoms like long-term COVID, or are you taking any people post 6 months that are 6 months post-infection?
Yeah, the interesting thing is that we have two kinds of data, generally, not only us. We have data from people that we saw people regardless of whether they reported symptoms. So we found complement dysregulation even in people that didn't have symptoms at all. So they had differential abundance of complement proteins, but you couldn't really say anything more about it other than it looks like, Alzheimer's. And that was probably even worse in people that had verified and lasting cognitive impairment. But there are studies that, other studies and probably larger ones from cohorts that had people post-ICU and people that had, let's say, definite deterioration, that showed that complement, there's a paper in Science, I think I could cite, that showed that complement remained, let's say, the premier culprit. And the paper in Science, I think, in some of these patients suggested that immunothrombosis might be an ongoing event for some of them. So yeah, that's really all we know. We do need a meta-analysis on the proteomic studies, even if it's going to be very hard.
Thanks a lot for the talk and answering this question so clearly. Any other question for George before? Okay. Thanks for your patience. Thank you all for joining the webinar. Just to let you know, we will have our next Instruct webinar happening on the 19th of November at 11 a.m. CET, and it will be 3 speakers from Lithuania talking. And hope to see you soon for our upcoming webinars. Have a lovely day.